funding. So that's also a bit of a frightening thought. So I'd like to quickly move on. I'm delighted that John Hickey's agreed to come and talk to us today and look a little bit beyond just the sort of sequencing we do now, but where we might go in the future with our Bronto bites and whatever came after Bronto bites. Thank you, John.
Thanks, Graham, and thanks for the invitation to speak here, and also for you, the audience, for coming to attend. So before I start, I'd like to address just some of the issues perhaps that Bob was talking that are a little bit different with what I'm about to present. So firstly, there has been a number of references in the conference about the price of sequencing, and my assumption is based on a paper that I saw published 2 years ago where the cost of sequencing was considerably lower than what is perceived to be the true cost at the moment, at least. So that paper said that you could get a DNA library, which is an important part in the sequencing step, for $15, and that you could do a 1x sequence for $120, which means you could get a 0.1x for $12, which would make the price of sequencing in the way that I see or think about the way that we should do it competitive with what we can do with SNPs. So that's the central thesis to all of this. So I guess my talk is a little bit, I'm going to have a review of what we might call genomic selection 1.0. I might talk a little bit about the properties and promises of genomic selection 2.0, discuss a bit how we might generate the data that we will need to do to deliver genomic selection 2.0, perhaps discuss some of the issues that Grant was discussing about how we might benefit from having this data, and I've already discussed what my overall hypothesis is, well, perhaps I haven't, which explicitly is sequence data is really powerful, but it's only really powerful if we have huge volumes of it. 1,000 or 10,000 sequences will be of little value, but a sequence on 1 million animals will be really powerful because we'll be able to do really good fine mapping. That's the sort of hope, at least. So I'm going to break genomic selection into 3 perhaps artificial categories because they perhaps don't exist. So we could propose that the original model could be genomic selection 0.0, which was that the predictions would derive from linkage disequilibrium information. What has happened in practice is that genomic selection has been the prediction accuracy has been driven by linkage or co-segregation information. But in the future, perhaps with massive data sets, we might be able to get the predictions to be driven by LD or, in fact, hopefully directly from the causal variants themselves. But of course, that will require lots of data. So obviously, genomic selection 1.0 has been a major success. Obviously, accurate breeding values are possible. We have a shorter generation interval. The dynamics of how it works are well understood. We have good systems in place to do that, to deliver it and implement it. But I think most importantly for the future, breeding programs have the capacity to generate really lots of genomic information, and we could continue to generate that genomic information in a way that is compatible with genomic selection 1.0, or we could change the way we're generating the data and start to do it in a way that might give us greater opportunity for benefit in the long term. So while genomic selection 1.0 has been powerful, several speakers this week have basically alluded to this same problem that I'm going to refer to here, that the predictions, because they're driven by linkage information, they don't persist very well, and therefore we're quite limited in what we can do in some senses. And here we've got some accuracies, if we like, on this column, and those accuracies can be high or low, and those accuracies, the value of those accuracies is entirely dependent on the number of close relatives you have in the training set. So if we take this black line here, what essentially we want to do is pivot it on this point and then move it all up, so that we would end up with extremely high accuracy for all individuals, as is depicted by the red line. And essentially, I think the only way we can do that is really fine map large volumes of the causal variants. Imputation has been central to genomic selection 1.0, and several people have been working on algorithms and strategies and genotyping platforms to ensure that we can get really good, what we might call imputation accuracies over in the far right column or the far left column, and at reasonable prices. So we were able to genotype lots of individuals and get what we consider to be good accuracies. But in fact, the information available to do imputation is quite low. So not all markers on our low-density chip are informative for all SNP. And it's quite surprising when you actually go and look and see how many informative markers do we actually have. So this was just one data set that we pulled out, which had 23 low-density markers on one chromosome, and only 10% of those markers were actually informative for imputing either the paternal gamete or the maternal gamete. So even though we have something like 700 markers genome-wide, we have very little information for doing imputation. And despite that, our metrics of measuring imputation accuracy tell us that we're doing very well. We're getting a correlation of 0.97. So we think we're doing really well, but I think actually the information is okay and sufficient for genomic selection 1.0, but it's not going to work for genomic selection 2.0. So I think we perhaps could change our thinking. We can accept that sequence information is falling in price, and the prices that I quoted at the start of the talk suggest that it's now possible to think at least that we could have sequence information on everybody. So I'm going to talk a bit about how we're going to generate that sequence, and then perhaps how we will benefit. And just maybe this is a bit of a silly thing to say, but I actually think at the next World Congress, well, what I've written is that we will have some people presenting results with 1 million animals with sequence. What I actually think is true, somebody will have 500,000 animals with whole genome sequence. But I upped the number to 1 million because I thought if you think back to where we were 4 years ago, nobody thought we would have the size of data sets that we do today, so I had to increase the number a little bit. So with such a data set, I think we can do what we might call industrial scale fine mapping, where perhaps 50% or some large number of the total genetic variance could be mapped to its causal variance. Under those circumstances, we could do several things. The first thing we could do is operational simplicity. So if we have full genome sequence on all our individuals in the nucleus, multiplier, and commercial layer, for example, or some degree at least in the commercial layer, we have a single genetic evaluation for all lines, all breeds, etc. So we have some level of operational simplicity. When we have persistent predictions, we can make accurate predictions in the multiplier layers, we can make accurate predictions across breeds, and perhaps there's an opportunity here also to take commercial data, use that as our training set, and then use that training set essentially to make selection decisions in the nucleus, but not doing any phenotyping in the nucleus, for example. I actually think sequencing can be cheaper because you have lots of opportunities to play with the parameters of sequencing that you don't have to play with when you're working with SNP data, because you tend to get locked into SNP genotyping platforms for a considerable period of time. They're less flexible. But we could also be a little bit more kind of, if you like, pseudo-science science fiction. And these are just a list of things that I thought of. I'm sure there are many other things that one could think of. But one thing is, as I consider it at least, in genomic selection 1.0, we're not making explicit use of de novo mutations that I believe we were implicitly using when we were doing progeny testing. And perhaps we're not seeing a negative effect of that in the short period of time, in the next few years, but in the longer term, we might start to really have some problem with that. I'll talk a little bit at the end of the talk about how we might use higher rates of recombination, how we might use genome editing for complex traits. I won't talk about how we might use sequence data to rapidly respond to novel disease outbreaks in herds, for example, or how we might use greater biological understanding to essentially to be able to select outside of the constraints of a genetic variance covariance matrix as it currently exists. But broadly speaking, it's just about better utilization of our genetic variation and better monitoring of it. How is our variation being created and utilized in our populations? And there are probably other things we could do. So how will we generate this sequence data on everybody? Well, obviously, imputation will be central because it's going to cost an awful lot of money if we don't use an imputation approach. So we could use a standard imputation approach, and actually this is the title of my World Congress talk from 4 years ago, which is essentially proposing that we could use a standard imputation approach to sequence a small number of sires at really high density and impute that data into the rest of the population. And I actually think there's now a better alternative because the technology has evolved and people have been thinking creatively in other fields, particularly human genetics fields, about how you work with sequence machines to give us new opportunities such that I actually think the top way of, or the standard approach is very suboptimal for what we want to achieve. So essentially, the idea is that rather than sequencing everybody at high coverage, or a small number of individuals at high coverage and imputing that data, we would be much better off if we sequenced everybody at low coverage. And as somebody, I think Bob earlier said, if we take a scenario where we have, let's say, 30 individuals who share the same haplotype, and we sequence those 30 individuals each at 30x, that means we're going to sequence that haplotype 900 times in 30 individuals. And that's, I think anybody would agree, that's a kind of a real waste of resources. So it's much better to sequence essentially that haplotype 30 times, but spread across perhaps 90 individuals. And if we do that, we can use the sequence data on both sides of the imputation task, if you like. So there's one aspect of imputation, which is we need to resolve what haplotypes are present in our population. And then the second aspect of imputation is we need to determine which combination of those haplotypes each individual carries. The standard imputation approach would only use the sequence data for the first part of that, and you would get a considerable penalty in the second part. And this type of approach of having, it's a bit like a big data approach, which is common in lots of science, where we essentially have we recognize we can collect an awful lot of data very cheaply, but that each data point in that data is quite unreliable. We use algorithms to recover the quality, and then we benefit because the total volume of the data overcomes any individual weakness. So just to pursue that a little bit further, I need some definitions. So obviously X is the number of reads at a position, so in sequence data, such that 1X is 1 read and 2X is 2 reads, etc. And I think Jerry Taylor said we all need 25X, and that's true if you're thinking in isolation, but if you're thinking at the level of the population, I would argue it's not true. So if we take I'm not sure there's a mouse here, Graham. Is that? Yeah. So if we take this individual who has these haplotypes, when we do the sequencing, we essentially fragment the DNA and then read these pieces, sort of. So if we're going to focus on reading these pieces here, a 1X read we would read the green piece or the red piece. If we did 2X, we could read the green piece twice, the red piece twice, or the red or the green. And the more times you read, the better chance you have of reading both the red and the green piece. And it's important to be able to read both of the red and the green piece if you want to determine that, for example, at this locus, this individual is heterozygous. And if you're sequencing in isolation of your population, then of course you need to do high coverage sequencing. But just to clarify, there's also different ways, besides manipulating X, that you can also think about lowering the cost of sequencing. And the most common, at least to my knowledge, the most common of those is genotyping by sequencing, where essentially you get restriction enzymes, and you chop the DNA at sort of random positions along the genome, and you can have different restriction enzymes which chop the DNA at more or less frequently, and then you can get your sequencing machine to focus its sequencing effort on the small pieces of DNA immediately adjacent to these restriction enzyme cut sites. So effectively you have a way of having a certain number of markers randomly distributed across the genome, and with different restriction enzymes you can have any number of those markers, be it 3,000 or 3 million. And then you can sequence those markers as many times as you like, so you can have 3,000 markers at 10x, or 3,000 markers at 1x, or 3 million markers at 10x, or 3 million markers at 1x. So that's why there's lots of ways in which you can play with your sequencer to affect the costs. So a little example of how this low coverage sequencing approach might work. So we have this sire family with the sire having these haplotypes, who produces these 4 children. They get DAM haplotypes that are depicted here. If we did 8x sequencing of these individuals, we would have the sire's haplotypes here, and we're ignoring the DAM haplotypes, but we would have the gametes of the progeny, or the sequences of the progeny, depicted here. So if we use the pedigree information, which is essentially what we do in imputation, we would just basically stack these haplotypes such that we turn the sire from having 8x to having 24x at no extra cost. And I think that would be a sensible thing to do. If we were doing this with GBS data, what we would essentially be doing is sequencing subsets of the DNA. So we would get this piece, but we would have the white pieces missing, for example. So perhaps a much better way to do it would be some sort of random distribution of the sequencing across our individuals in a pattern such as this. We could then take this data and derive consensus haplotypes from it, and then use the data again to impute these consensus haplotypes back into our individuals, and we could get quite effective imputation. And if you look at why we get quite effective imputation, it's because we have information which is very close to the recombination locations that enables us to really fine map where we are doing the imputation, if you like. But of course, just as it has been in genomic selection 1.0, you can afford to spend more money on the genotyping of an individual who makes greater contributions to the population. And for example, we were able to genotype sires with more density or more expensive chips than we could genotype dams, and we could genotype dams with more expensive chips, so we could genotype individual selection candidates. And it's the same thing here. While I would argue that we don't really need to do anything other than sequence the candidates at very low X, it probably does make sense to think that we would do a candidate, for example, at 0.01x. When that candidate becomes a sire, it could be done at 4x. If it became a dam, it could be done at 1x. And if it became a grandsire, it could be done at something like 10x. I think that would probably, some strategy like that will probably emerge as being the best. So we do have an imputation algorithm that is designed for this sort of data, but I'm not going to talk about it other than to say it's not ready for use, but that it is giving good results. But we have been doing some simulations to evaluate the overall strategy, and for doing that we've been using Beagle. And here we're going to compare a standard, let's call it, if you like, 1,000 bulls high coverage sequencing of a small number of individuals approach with an everybody at low coverage approach. So we have a total of 1,000x sequence. We can distribute that across 25 sires at 40x, and we genotype the progeny with 200 SNP, with the objective being we're going to impute this information in the progeny. Or we could distribute that 1,000x sequence across all of the 1,000 progeny, each at 1x, and then impute high coverage sequence in those progeny. And that's scenario 1 and 2, and you can see immediately that there's quite a benefit from doing it the second way. So another reason why, so probably a large part of this benefit comes from our being able to better fine map the recombination locations. But another reason why the standard imputation approach won't work with high coverage sequence data is depicted if we compare scenario 1 and scenario star. So they're effectively the same scenario in that the sires are being sequenced at some x, and the progeny are being genotyped with 200 SNP. But in scenario 1, the sires are being done at 25 or at 40x, whereas in scenario star, they're being done at infinite x. And you see there's quite a difference between these two situations, and that's because even if we have 40x, we don't have even distribution of that 40x across the genome, such that we have some regions that get really underrepresented, and they contribute quite heavily to imputation accuracy. So anything we know about sequence imputation or genotype imputation does not necessarily apply to sequence imputation as we go forward. So this is perhaps a little bit of a silly idea, but I just thought it was interesting. There are other ways to think about how we might be able to increase our imputation accuracy when we have sequence data. So if we think about sperm cells, they're actually haploid, such that if we were able to get the sperm cells of a sire and sequence them, we would have the sire's haplotypes effectively phased, and phasing is a major component of imputation tasks, so we would actually potentially benefit from that. So again, now we have 1,500x in total, so we could do 30 sires, or we could do our sires at 30x and have the progeny with 200 SNP. Alternatively, we could say we're not going to sequence the sires at all. Instead, we're going to sequence 30 sperm cells of each sire, each at 1x, and again genotype the progeny with SNP. And if we do that, we immediately see we get quite a big bump in imputation accuracy. But it's still not perfect, and there's a little bit more that we can do. So if you think about the way a sequence machine, when it samples the data, it's taking something from either the paternal gamete or the maternal gamete at each position, independently of what it took at the previous position. And those reads are very short, so it's constantly sampling the sire's gametes in this case. And that's a good thing. But if we were working with sperm cells, you essentially sample whole half chromosomes, and then you get another whole half chromosomes. And if you're only working with 30 sperm cells, you can end up with quite a bit of the genome not represented, which is why there's a difference between here and here. So the way to solve that, if you like, was to add some amount of sequencing on the sire to do this sampling very well, and then some amount of sequencing on the sperm cells to represent the haplotypes quite well. So that turned out to be a very good option. And these are all imputation results in Beagle, and you see we're already getting really good results with quite a simple imputation algorithm. So we would accept that we don't have this infrastructure in place in the animal breeding community to implement this low coverage sequencing approach, at least the imputation of it. But nonetheless, we thought it might be useful to evaluate how well would that sequence data compare to SNP data for achieving what we currently achieve today, which is, let's say, genomic selection 1.0. So we have a simple simulation where we have, this is GBS sequence data. So we're going to have 3,000, 10,000, 60,000, or 300,000 markers. Those markers can be at any x, sort of from very low up to 20x, and ultimately can be SNP, and if they were SNP, they would effectively be infinite x. And then you see what is the asymptote of prediction accuracy. So we have some training set and some set of selection candidates, and the breeding values of the selection candidates. What is the asymptote? And as we get to higher marker density, the asymptote gets to be surprisingly low. So this is only 300,000 markers, and it's well within the price range of what we currently pay for SNP data in livestock to have 1.1 million markers at 1x. And then we would imagine that without any imputation, we would be at the asymptote of prediction accuracy, such that we can achieve what we need to achieve today, but in the longer term, we can have really good imputation of sequence data to generate these big data sets. And then you can also think about other ways to derive benefit. So if we have, let's say, a fixed volume of sequence data, so focus on the green lines only. So we have 10,000 X at each position. So what you could do is you could do 500 individuals, each at 20X, or you could do 50,000 individuals at 0.2X, or any number in between. And then by doing that, you manipulate the size of your training set if you have the, this phenotyping capacities to do this. And overwhelmingly, the right thing to do was to have lots of individuals, each with poor genotyping. And we also did the same thing with the prediction set of individuals. So again, manipulating the selection intensity. And again, in fact, for the prediction set of individuals, response to selection was even more, let's say, the result was even more obvious than here, for making more candidates, each with lower accuracy. So now I'm going to talk very briefly about two ways in which we could derive benefit from having huge volumes of sequence data. The first being genome editing for complex traits. The second being inflating recombination rate. So genome editing is, if you like, it's a technology that's been around for a while, but it's now getting to the stage in which it is almost feasible to be used on an industrial scale. At least it might be in the next few years. And it's simply a process that you can use to add, delete, or replace nucleotides in an individual. So much of the focus of genome editing to date has been on simple traits. For example, my colleagues in Roslin have created this pig they call pig26, which has a single base deletion in one of its genes. And that made some newspaper headlines in the UK last year. But we're focusing here now on the application of genome editing for complex traits, so assuming that in the future it will be cost effective to edit sires for some number of alleles, and then trying to evaluate what would be the benefit. So we think about genome editing in this context as effectively controlled recombination, and by controlling the recombination better, you can move the variation in your population around faster and therefore make more genetic progress. So we have a simulation where we do selection, and the selection is on true breeding value. So it's effectively genomic selection with perfect accuracy. And under those circumstances, we get the green rate of genetic improvement. This is genetic gain here for a trait with 1,000 QTN or 10,000 QTN. And then we say we're going to edit the selected sires for the 20 QTN that they of largest effect size that they are not already fixed for, and you see you get quite a bump. And these numbers may be unappealing, and you might sort of say, well, we can't do GWAS for these type of numbers. But I don't have time to go into the details other than to say the numbers stack with what the results of the current GWAS people are getting. For example, the Peter Vischer papers from this morning, where with the kind of numbers that he's getting with his GWAS of this size, fit within the range of what we would need in order to achieve this. So when we do genomic selection 1.0, we think about the accuracy, and it's deriving from this correlation between SNP and marker. So in our case, it's LD, or a linkage at the moment. In the future, it might be LD. But either way, you're depending on some correlation between the markers and the QTN. If we had massive data sets and we were able to have the QTN directly driving the predictions, we wouldn't be depending on those correlations for our prediction accuracy. So if we were under those circumstances, we could actually inflate recombination, and if we could inflate recombination, we could release more genetic variation in each of our generations. So if we think about, we have an idiotype which would have haplotypes with the big Q and the big A. That's what we want to achieve. But if we start in our population with haplotypes like this, it doesn't matter how many rounds of selection we do, in the absence of recombination, we can never achieve the idiotype. So there's something useful about recombination for making a response to selection, obviously. And if we think about domesticated pigs have got a higher rate of recombination than the wild pigs, and in plants, the progenitors of domesticated plant species have got a considerably higher rate of recombination than the wild species that have never been domesticated. So there's quite a bit of evidence to say recombination is useful. So most of our standing genetic variation is trapped because of this limited number of recombinations. So if we have higher rates of recombination, could we release more of that genetic variation in each generation? So recombination is manipulable. It's under environmental and genetic control. It has a standard heritability. Causal variants have been found that increase recombination by 15%. For example, this is the result from Decode Human Genetics population from last year. So we could do genome editing for it. We could select on it, or we could do some environmental manipulations. So we did a simulation which has several generations of selection. We have a grid, if you like, of genome lengths and a grid of selection intensities, and then we found a pair of scenarios which utilized genetic variation at the exact same rate. So that's, I think, a fair way to compare two different breeding programs at the rate at which we're exhausting genetic variation. And in this case, it was 10 Morgan genome length with a 5% selection intensity, or 100 Morgan genome length with a 2.5% selection intensity. And we were able to make more genetic progress by inflating recombination, and there are more details on this in Gabor Mazzara's talk on Friday. So with that, I would like to close by saying that I think, yes, we need to generate huge amounts of data sets, or huge data sets with lots of sequence and phenotyped individuals. All of what I have said depends on us being able to map a large amount of the quantitative genetic variance directly to its causal variance. I think this is the pig session, so I think if you like, dairy breeding or the dairy geneticist, that has been the poster boy of genomic selection up to now for genomic selection 1.0 because of the favorable sort of industry structures of dairy breeding. I think genomic selection 2.0, if driven by this type of data set, is going to require massive investment, and large pig and poultry breeding companies may have the capacity to do that, and therefore perhaps pigs and poultry will become the poster boy for genomic selection 2.0. Thank you.
Thank you very much, John. That was terrific, and I like that idea. When you were talking about 1 million individuals being sequenced, I know I've misunderstood what you said, but I think you've set the target, the equivalent of putting a man on the moon in the next 4 years, we're going to do. And you heard John say we need to be the poster child next time. So there's the challenge. Moses.
Just have a question in maybe follow-up, but Bob mentioned that we need to genotype at least some folds to be able to have a good sequence. I think when we need to do imputation, we need to construct the haplotypes correctly to have an accurate estimation of our, do good imputation. But when you say to do one fold, I think we will have lower accuracy on the haplotypes that we generate. That one will probably, I think, should cause that our imputation accuracy goes down. Is that right, or is it?
No, I actually don't agree with you. I think we have done simulations which show that the other opposite is the truth, and it actually is logical from an imputation perspective that the opposite would be the truth. So I actually disagree with you.
But your haplotype estimation will become incorrect then.
But you're thinking in terms of an individual in isolation from the population. Okay. We need to start to think in terms of sequencing a whole population, not sequencing an individual. And when you do that, you don't want to sequence a haplotype 900 times, because that would be a waste of money.
Okay. Thanks.
Thank you, Hans.
Hi, John.
Hi, Hans.
Thank you for a very thought-provoking presentation. My question is along the same lines. I agree that we don't have to do 25x, and basically we've shown that in 1,000 bulls. But have you considered sequencing error in your simulations, and how do you think that would affect your results?
So we haven't considered sequencing error, but I still think the results still hold. And yes, I think the benefits of doing it this way far outweigh being able to sequence a small number of individuals very accurately. That's just my view. But I'm aware that there's a lot of opposition to this view.
John, a really simple question. Is there an intermediate optimum for recombination rate?
So, Rob, yeah, there must be, because in almost all species that I've been able to get information have a genome length of between 20 and 40 Morgans, so there must be something fundamental about that level of recombination to protect the species from dissolving or something. So yes, but let's see. I'm aware that in the UK there is a research project in plants where they're actually trying to quantify recombination and manipulate it to evaluate this. So I think it'll be interesting.
John?
John Bastiaanse, Wageningen University. Thank you for showing the most exciting ideas I've seen this week. I had a question. At some point you said for genomic selection 2.0, 95 to 98% accuracy is not good enough for imputation. And are you fixing that gap? Or what do we need to do to fix that gap up to 100%?
So that was the one sentence in my talk that I was a little bit uncomfortable about saying. But it did fit with my story, so I thought nobody might ask me this question. But I'm a bit blanking now, but sorry. I'll talk to you later about that. Okay. Yeah.
JP.
Related to the question on haplotyping and sequencing error, I think we are all looking at where this is going in terms of cost, but we have some prospect of where this is going in terms of length that we are able to sequence and sequencing error of that. And that's changing very rapidly. Do you have any insights on that? Like able to sequence such a long?
Well, that's why I don't, but there's two things I could say. We can start to think about sequencing sperm cells, so then you get really long reads, and you can also sequence pieces and put, apparently, I don't know much about sequencing beyond superficial things, but apparently you can put a space between the sequence, so it effectively becomes a long read, and you have two SNP at either end of the read and a big gap in the middle, and then you can phase these two SNP, and ultimately you can stick the whole genome together in that way. So that's basically.
So you envisage that within 5 years' time there will be data sets where there's millions of animals sequenced, so probably we'll have millions of SNPs on those animals. You touched upon the imputation process. What's your vision on the genomic prediction models we need to deal with all this?
So I looked, so obviously we're going to have a dense system of equations, right? And we're going to have, I would guess, let's say 40 million things we need to estimate. And I looked up the world record for systems of linear equations, and the largest system that's ever been solved is 11.8 million effects. And they used a supercomputer, and one of the metrics for that supercomputer was the amount of electricity it used to solve the equations, not the time. So yes, I think we have a considerable computational challenge. But I spent 4 years in an office with you, Mario, so I'm pretty sure you're going to solve it.
Good answer, John. Max?
Yeah, very interesting talk. I want to kind of bring it back to something that troubles me in the industry, and maybe Bob can comment too here. But when I was swine genome coordinator, we had a big discussion about moving from the 60K chip to the 600K chip. We couldn't generate enough money to do that. The industry didn't have the interest in spending that kind of money. So bringing us back to reality here, combining both Bob's talk and yours and perhaps mine to a little, we have three enormous challenges, and they're all related to money. Better understanding of phenotypes in biology, more computing power, and the wherewithal to find the money to spend on molecular biology. Who's going to put this together? We have an industry that frankly is a bit conservative from a financial standpoint.
Well, I said I suspect someone is going to have 1 million, but I'm 100% sure someone is going to have 500,000, and I think I know who that's going to be. We have a way to do it, I believe.
Well, we don't have a comment. I make a comment in a minute. Last question, I think, and then we'll take a break. Nope, not you. You've gone most of it.
Very interesting talk, John, and I totally agree with you. It's like in the QTL mapping time when the plants, they have the same plant breeders, they can have the same genotype multiplied in the field. And then when you have a single animal with that genotype, they say, you don't have replication, but you are replicating across individuals. So I agree with you. I think that's the way to go. But it's clear that the constraint will be phenotyping then, right? So we are going to have the genomes, and then we want to improve for disease resistance, and you can measure, 2,000 animals. Right?
Yeah. Again, it's the same answer as before. I agree with you, but I think we have a way to do it by phenotyping in the commercial layer. If we can make genotyping cheap enough, we can maybe do that. But that's,
Very good. Thanks, John. Well,
last thing, Graham. So we were talking on Twitter. I don't know if you're aware. There's a Twitter thing going on in parallel, and we decided we're going to have an animal genetics discussion group hashtag on Twitter. So we said we would launch that. So just telling everybody.
Well, that's a great idea, and I would encourage you to do that.